Okay. What about, we hear you, hooray. Okay, good, great. Okay, folks. No idea, sometimes it works, sometimes it doesn't. I'll just share my screen again. Okay, can you guys see that? Okay, righty-ho. Okay, so I'll just give you a quick, I mentioned about the midterm one last time. I'll just give you a few more details about that, and I'll send out an email as well. So just to be perfectly clear about this, right, if you look at the syllabus, it talks about midterm 2 covering lectures 10 through 17, okay? Now, on the syllabus, it reckons that we're starting chapter 31 by now, but we're not. So it will only cover up to what we cover in this lecture, which will be chapter 30. Okay, so these are the chapters. So there are a few chapters from Dr. Spagnolo. If you have any questions about those lectures, you can email him, you know, maybe arrange to meet with him or something like that. And then, you know, obviously I have office hours this afternoon from 2 to 3. So you have any issues over the lectures that I've done, you know, you're more than welcome to ask about those as well, of course. If that time doesn't suit you, just drop me an email and we can figure something out. Okay, a few folks have asked about, you know, what to focus on. Remember, and it's clearly stated in the syllabus that, that, you know, we're only going to examine you on stuff that is covered during the lectures. So if, you know, there's a lot in each chapter, and if there are some bits that are skipped or not mentioned, they won't be included. It'll be what we cover in the lecture. And occasionally, you know, stuff in the lecture is additional to the chapter as well. So sometimes, you know, there's other stuff that possibly, hypothetically, might be in the lecture that wasn't in the actual chapter. Okay, so it's the lecture content that's the important stuff to focus on. With respect to my specific lectures, people have asked about, you know, notes, etc. I put quite a lot of summary notes at the end of each slide that you can sort of take as a starting point for the structure, you know, if you want to use it for revision purposes. You know, of course, not everything can fit into 2 or 3 slides, so it's the entire lecture content, but it gives some kind of a, you know, a breakdown or some sort of structure to the overall issues that we've looked at. I will be creating the exam on Blackboard rather than Achieve this time. Lecture 10 is not up on Blackboard anymore. So you're talking about the recording, Said? Because did you email about that? Yeah, because I think Dr. Spagnolo said that when you go, I did actually. You have to filter it, yes. So when you go into Blackboard where the lecture content is, let's see if we can actually have a look at that. You have to, let's just change the date in the recording in range. Yeah, you have to click through the different, not all the lectures are on the front page that comes, you have to flick through them. Yeah, change the date. Is today lecture 17? Whatever it says on the syllabus, which is lecture 17, yes, but we're not talking about deuterostomes. We're still finishing chapter 30, okay? We're a lecture behind, basically. So, yeah, look at the syllabus. The syllabus is on Blackboard, okay? And write down these chapter numbers or lecture numbers. Okay. So it'll be on, and I've created an exam folder, okay? And you can see that right here where I've got the pointer right now. I've created a new folder there, okay? And that is where the exam will appear, okay? I put in just a placeholder in there yesterday. I created that last night and said that the exam will appear on there at 12:15 on Tuesday. I think I'm actually going to put it on at noon, okay? We can start the exam anytime from 12 to 2:15. I haven't decided when I'll make it visible until, I think I'll make it visible until 1:30 probably. And I think the way you've got more experience of doing this than I have in Blackboard in terms of taking the tests, I think you can start it at any time, okay, within that window, as long as you start it within that time. I do not recommend leaving it till later, but at noon on the dot, there's sometimes an issue with, 246 people logging in, so that can create a traffic jam as well. Will the test be multiple sections on Blackboard? So like 4 sections of 14 questions. I don't know about that, Diana. Now, did they do that in 105, right? With that 4 sections, so 4 separate exams? Yes. And I think, is that a preference for people? Because I know that if it crashes and you have to restart, you just have to do that 1 section. Yes. Okay, it's helpful. Yes, yes, yes. Okay, so I will break it into sections. And in 105, did they just have them all appearing at the same time? Somebody says no. Oh, they appeared at different times. Yes, they appeared at different times. Next part appeared after you. Okay. I will chat to Dr. Holtzman about that and check how she did it, okay? I will try and do something similar if I can figure it out. Yeah, you need to go in order, yes. That's obviously beneficial because you guys are remote, right? I will chat to Dr. Holtzman about that, exactly how she did that, and I will try and set it up like that. Either way, I'll let you know, guys know, okay? Okay, there will be 56 questions in total. The total allocated time will be 75 minutes, so I'll break it up accordingly. Backtracking not allowed, I'm afraid. I know that upsets people because that's, when we're all there and you can be moderated, then that's obviously a good way to do it, getting all your questions that you know down initially and then going back. But we cannot do that for when you guys are remote, I'm afraid, okay? It is better in parts because we can take a bathroom break. Okay, okay, very good. 75 minutes. Students for the accommodation letters, okay? Now, I have a few, and I have confirmed today by email for the few people that I have accommodation letters to say, I know about your accommodation letter and I'll adjust your time accordingly, right? There's one person who has asked for an accommodation, but I need the letter, okay? And I've emailed that one person as well. If there's anybody that thinks they're on accommodation and I have not emailed you about it to confirm that I will get it, you need to get back to me ASAP with your accommodation letter, okay? I'm assuming by this stage of the course, I know about all accommodation letters. But it's possible that some people might have only told Dr. Spagnolo and not me. He forwarded me his. At what time will the exam close? At the end of the day? No, it'll be sooner than the end of the day, okay? I'll let you know about that once I've decided. What do you mean by randomized pool? So the questions don't appear. What you see will not be necessarily the same as the order, etc., of the way that other people see it. Not like Achieve, right? Achieve was like a dumpster fire. Okay. I don't know because I didn't do the Achieve exam. But I'm guessing you guys, so you've done 105, so it'll be similar to that, I guess. So what I'm saying is question 1 that you see won't be the same as question 1 that somebody else sees. Okay, so people prefer Blackboard tests by the sounds of it. Oh yeah, yeah. Okay, good. I do as well. It's a lot easier from my perspective as well. Blackboard is, Achieve is really confusing. Okay, good. Makeup exam. I've had one request from somebody. I need that, letter, and then I'm happy to arrange something else. But I've only heard from one person, if you have a doctor's appointment or something and a valid documentation of that sort of thing, tell me sooner, not like 2 minutes before the exam. How you're splitting the chapters into sections. When you figure it out, you will tell us how you are splitting the chapters into sections. I don't understand your question, Melissa. When I figured it out.
So I was saying that there are like I think 6 chapters on the test. So I was wondering, does that mean that if you do like a few chapters, it'll be, I mean, few sections, there'll be like 2 chapters on each?
Two
Like, will you
chapters on each what?
will you be splitting the test into sections of like 2 chapters? Is what I'm saying.
I will, if I split it into sections, I'll probably randomize questions across sections.
Oh, okay.
Okay. Priest professor did lecture 1, 2, quiz 1. Yeah, she might have done that. I might necessarily not do the same. Okay. I might randomize questions across sections. Okay. I'll see how it breaks down. Okay. Any other questions on the exams before we get going? If you've got any, detailed questions, you can email me or, what's the word? Office hours.
Is the syllabus like? Is it the same format? Like, could we still use the syllabus as our guide for the dates?
Dates, yes, but some of those chapters have flicked around, right?
Okay.
Yeah. Do you want me to? Would it be helpful if I updated the syllabus? Would that help?
Yes, that would be great.
Yeah.
Thank you.
Because I haven't quite figured out. I'm still deciding what to do with makeup time. This is not atypical. Dr. Baker often gets behind them, and he does one of two things. Sometimes he really goes quick for some lectures. Or sometimes, we have the option, I might drop one of the chapters because it's one that people have done 105. I know somebody said they haven't done 105 yet. Yes, there is on there. Did Professor post the slides? Yes. So the slides are the same as the ones I posted the other day, but we didn't actually get through this PowerPoint yet. Okay, and I need to get going on it now or else we won't get it through today either. Okay. So exam 2, 100% basic lectures. Yes. Okay. I'm going to move on now, okay? So just a reminder of where we are in the grand scheme of things. I really like to zoom out and zoom back in again because it's easy to get lost in all of this stuff that we go through, right? It's like a Russian doll. When you go through these phylogenies, you get to the end of one phylogeny, but then actually each one of those branches opens up into a whole new one. So it's good to remind ourselves where we are. Since I've been taking over lectures, we've been talking about the animals, okay? We initially talked about basic features of animals and some of these organisms here, the sponges, the ctenophores, placozoans, and cnidarians. And then in the last couple of lectures, we've been talking about those animals that with bilateral symmetry in their common ancestor. And we've been talking about the protostomes, okay? That group of organisms that's characterized by the blastopore developing into the mouth, except for arrowworms are a bit of a weird oddity with that. And what we're going to talk about this time, so we talked about the lophotrochozoans. This time we're going to talk about the ecdysozoans, okay? So that expands out into this red area here. We're going to focus on these groups of animals here, finishing off with the arthropods, okay? The most diverse group of animals on Earth, okay, and some of how they evolved and some of the reasons for their incredible success. Okay, about 1.25 million species of described arthropods compared to about 1.6 million in total of all animals. So what is it that characterizes the ecdysozoans? And the key thing that characterizes them is the cuticle that they have surrounding their body, okay? You can see if I go back to the phylogeny, this is a synapomorphy of the ecdysozoans, something that was in the common ancestor and it follows through with these organisms, of the exoskeleton molting, it's called there. So they're characterized by this external cuticle that surrounds them, that is secreted by the epidermis, the outer layer of skin. And the cuticle provides some degree of protection. It differs depending upon what type of ecdysozoan you are, and also a degree of support. So it can act like in some cases, a sort of external skeleton if it's been made rigid. So again, it differs between the different groups. Now with this cuticle though, in order to grow, it can't grow with the animal itself. It has to be molted and replaced with a larger one. Okay, and that process is called ecdysis. Okay, so they have an outer shell here, and this thing is growing, and so it's had to shed that cuticle, comes out of it, and for a short period of time, it's relatively soft, pretty immobile, so it's quite vulnerable to predators, and its cuticle rehardens, and then it's good to go again. So this is thought to have evolved about 500 million years ago, apparently only once, so it's an synapomorphy of the group. And so all ecdysozoans have a single common ancestor, and there's strong genetic evidence to support that as well. Some of that strong evidence comes from their Hox genes. If you remember, those are the genes that we learned about that are important developmental genes that are controlling developmental pattern along the body. And they're highly conserved in many organisms. That means they're good for looking at long evolutionary time periods. Does molting happen at the same pace for all species? I'm pretty certain no. But because some of them got extremely long lifespans, some of them have not. So it's not the same pace, no. Under the bugs and the spider. Okay. Answers to lots of bugs and roaches. There are a few bugs and roaches pictures, but I kept out, we're going to talk about elephantiasis disease in humans, and I left out the pictures of those. You'll be glad to know, Dana, because I can't bear to look at them myself. So Hox genes, good evidence of a single common ancestor. And actually there's evidence from fossils going back a long time where actually there is, some of these organisms have been fossilized in the process of undergoing ecdysis. Are butterflies ecdysozoans? Yes, they are. Okay, and we mentioned butterflies briefly, or insects at least. So when you think about ecdysozoans, they can, in a general sense, be broken down into two sort of general groups. Those that have a worm-like body form, and those that have limbs. So we'll talk a little bit about these, but you notice that these worm-like body forms, they're not all in a single clade. Some of them here, the nematodes and horsehead worms, are actually in this bottom clade here, but they're distinct from these other groups that have limbs. Okay. So let's just look at some of these worm-like organisms. So in these groups, the cuticle is relatively thin and flexible compared to these other groups here. Okay. So it offers a protective layer, but it's a fairly modest protection compared to other groups that have a much more thickened cuticle. And also that thin cuticle allows exchange of gases and minerals and water. So chemicals can diffuse across that relatively thin cuticle because it's relatively thin. And that has some benefits in terms of perhaps gas exchange, etc. But there are also certain limitations to that as well. One of the major restrictions or limitations is that they are restricted to habitats where they're not going to completely dry out, basically, because they have this thin cuticle, so water can evaporate across that surface. So they're restricted to wet or at least moist habitats. And then accordingly, many of them live in marine or aquatic areas, in sediments. In terms of their feeding, some of them are deposit feeders, That means that they can feed on organic matter that, for example, might have settled on the surface of the sediment. They can sort of pick that up, or actually eat sediment itself and digest out that organic matter. But some of them are predatory, so they're eating other animals. All animals are, well, animals are heterotrophs, remember. For that, they have a toothed pharynx. So the pharynx is a muscular organ that's at the anterior part of the digestive tract. So what we would sort of think is in the mouth region, basically. There are freshwater and parasitic types that absorb nutrients directly across that thin cuticle that they have. Okay. So let's look at some of these groups. So we're going to go through these. Just in general, when you look across these, right, you can see that quite a few of these groups are kind of very small, obscure, you know, you might think of as obscure groups because people don't often sort of know or hear about them. In fact, one of these groups was only discovered in the 1980s. But you see, like the priapulids, for example, 20 species compared to arthropods, 1.25 million. Nematodes, 25,000 is the biggest group within this. But these other groups are kind of small groups, basically. So let's look at this, these top three groups that are in this single clade here. It consists of the priapulids, the chinorhynchs, and the loriciferans. Okay. So these priapulids, there are about 20 known, or there are 20 known species, and worm-like things between 0.5 centimeter up to 20 centimeters, so maybe this big. I had the same question. Are they strictly restricted to wet habitats, or is it possible to find one in the desert? Desert, extremely unlikely, I'm guessing, when I look at these groups. These are purely marine, so we'll talk about these. So nematodes are the most diverse in these. They're very important in soil habitats, for example, but are restricted to, you know, moist areas of soil. So some of them in soils, etc., but I'm guessing not in a desert. Others are marine or aquatic. So these priapulids are all marine worms, basically. They form these burrows in soft sediments in the seabed, and they prey on other marine worms with this toothed pharynx that I talked about. They have this sort of curious behavior where they're in the burrow, and then there's this sort of part that comes out, and they sort of go around different parts of the sediment, and when they've got something, they then withdraw it back in again. These chinorhynchs and the loriciferans, though, these are much tinier little organisms. So the chinorhynchs, so less than 1 millimeter long. Same with the loriciferans, less than 1 millimeter long. So very small. Only 180 species of chinorhynchs, only 100 species of the other group. Live in marine sands and muds, and these are deposit feeders. So that means they're feeding on sort of organic matter, for example, of dead plankton, for example, that might have settled down onto the seabed. And this other group live in coarse marine sediments, so marine sediments as well. And as I mentioned, this loriciferan group, they were only discovered in 1983, so really recently, by a Danish scientist who actually discovered two other, this is a phylum of its own. He's actually discovered 3 separate phyla during his lifetime. So obscure, small group of organisms. So in general, these are all marine. Most are developed directly from an egg into an adult form, as did their fossil ancestors. So in other words, when they have some offspring, you know, a juvenile or a hatched priapulid is going to look like a small priapulid that'll get bigger by shedding its cuticle with time. These other two groups of worm-like creatures, the nematodes and the horsehair worms. So these nematodes are by far, you know, the most important in terms of being the most species of these worm-like ecdysozoans. Okay, and they also play an important ecological role in terms of soil health and things like that. So about 25,000 described species. Somebody asked the other day what described meant. That means a scientist has sat down, figured out what this animal is, what group it belongs to, and given it a name, right? But it's estimated that there are over 1 million, well over 1 million species. A whole bunch out there that haven't been described by science yet. Probably the most abundant and universally distributed of animal groups, because they're small and just like there are lots of them in soil and all sorts of different habitats. So abundant in marine sediments, lake sediments, soils, about 65 per square inch, and a rich farm soil apparently. And they're important for soil health in terms of regenerating organic matter, etc. Many of them are predators feeding on protists and small animals. Many of them are actual parasites as well, and really important parasites in some cases of either plants, so they can be an agricultural problem, or in animals, including humans. So an example of a human parasite is this thing called trichinella. So this is something that you get from, or you can get from eating meat that hasn't been cooked properly, especially pork. And so the way that you get this is that these nematodes, they form a cyst in human or in muscle tissue of whatever their host organism is. And then if that's a pig and we eat some pork and haven't cooked it properly, this thing will still be alive. And the larva is activated within our digestive tract. And that then attaches to the intestinal wall and feeds upon food in our intestines. And eventually they can bore through our intestines and get into the system and get into our tissues as well. That can be, either create severe illness or in a really bad situation, death as well. And another example is elephantiasis. That's the one I was saying I left the picture out. You've probably seen these pictures of, these poor people that have been infected by a nematode worm that causes this, and it invades the lymph tissue and the lymph nodes and blocks things up basically and causes amazing swelling and debilitating. And about 120 million people worldwide are apparently afflicted by that. So, some really debilitating parasitic infections, but there are lots of free living forms as well, such as those in soils. They're also a super important model organism. Somebody mentioned it in the previous lecture, C. elegans, okay, which is a soil nematode. And it's an important model species in the lab, and it's been really important in terms of genetic and developmental studies for a whole host of reasons. One of the important ones is that it's really easy to cultivate and it's cheap to look after and easy. You get loads and loads of them really easily. They mature within 3 days, so you can go through life cycles very quickly. They have a fixed number of cells, and those cells have been mapped really well. So if you look at an embryonic cell within a C. elegans and say, what is the fate of that cell, that's been mapped, and so people know what each one of those cells will develop into. And it's got a fully sequenced genome. It's I think the first or one of the first animals to be sequenced. So really good for studies like that. In Queen's College in the biology department, there are 2 labs you're probably familiar with, Dr. Savage Dunn and Dr. Melendez. They're both C. elegans labs, right? So super important model organism. These horsehair worms, this is a relatively small group related to the nematodes, about 350 species, a few millimeters up to 1 meter long in some cases, so they can be very long. And they live in freshwater, leaf litter, edges of lakes and streams, but they have a part of their life cycle. They are endoparasites, parasites that live inside a host organism of freshwater and insects. So this is an example of one. You can see this here is the worm that came from inside this cricket. So these things, like that stickleback cestode I showed you, they can grow really big compared to the size of their host. And like a lot of parasites, actually, they have these really interesting sort of behavioral modifications that are designed to be perhaps to the detriment of the host, but to the benefit of the parasite itself. So this particular one, it needs to lay its eggs in water, and it changes the behavior of the cricket so that when it's ready to lay its eggs, that cricket associates with water and jumps in the water and drowns, basically, so that the eggs will then be able to develop in water. Pretty cool. Okay, so how are we doing for time? Okay, we're good. We've been talking about the ecdysozoans, okay, the second major group of protostomes, the other being the lophotrochozoans. So we're talking about the ecdysozoans today, those animals that have an exoskeleton, a cuticle that they need to molt to grow. And we talked about that top group there that have a worm-like body form, okay? They're not all necessarily closely related. Some of these nematodes are in the same clade down here as these other ones here. Now we're going to focus on these organisms here, okay? That rather than being worm-like, they have limbs, paired appendages, which evolved about 540 million years ago originally. So this paired appendages is a synapomorphy of these groups here, okay? And then, so we'll talk initially about these first two groups, the tardigrades and the velvet worms, and then go on to look at the arthropods, which have another synapomorphy where the appendages have joints within them. So these two other groups, tardigrades and velvet worms, they just have these fleshy appendages. They're not jointed, like you'd imagine, you know, like a spider leg joint or something. So let's look at those. You can see that these again are sort of relatively minor groups. So 1,200 tardigrades, 180 velvet worms, compared to the most speciose group on Earth. He's so cute. Yes, I know. People do. Tardigrades, people always think tardigrades are cute, right? You know, their common name is a water bear, but these things are really teeny, right? They're only 0.5 to 1.5 millimeters long. Have no circulatory system or gas exchange organs. They're really small, relatively thin cuticle, dumping with legs. And so gas exchange can occur across that. And they live in marine sands and temporary water films on plants. And, you know, one of the extraordinary things about these is, a bit like the rotifers that we talked about last time, they can undergo this, this desiccation. In other words, if the water film on a plant that they're living in, for example, dries up, then they can dry out themselves mostly and remain there in a sort of a state of inactivity, virtually zero metabolic rate whatsoever, until, you know, it rains again or something like that, and up to a decade apparently. So a super long time. Okay. These velvet worms, now superficially, you might think they look a little bit like annelids that we talked about last time. Now remember, the annelids are lophotrochozoans, okay? So they are in that other group of protostomes we talked about that do not shed their outer layer in order to grow. But there's really strong evidence to suggest that actually they belong to this group. They're not part of the lophotrochozoans. And even though superficially they might look like annelids, when you look at them a bit more closely, they're not, compared to some of the features we talked about. If you remember, the annelids are segmented worms. These don't have that sort of clear segmentation. They have these fleshy appendages. The polychaetes within the annelids had little fleshy podia, but they had setae on the end of them. These have like little claw-like things. So there aren't different structures about them. I've never seen one of these things, but apparently they live in leaf litter in tropical humid areas. If you go online and look at YouTube, etc., you see that there's some cool videos of them, that they're sort of predatory creatures that have these special mouth parts to them that they can sort of shoot out. They're not very fast, but they can entangle insects, for example, and like goo them up, and so that the insects can't get away and feed upon them. What is the white spot? To have a purpose. You mean this white spot? No idea. That's I'm going to say it's a parasite, but the truth is I have no idea. It might have been injured or something. Okay, so those are the minor groups of this clade here that have paired appendages. Now we're going to talk about this group, so that have paired appendages but jointed paired appendages, right? And these are the arthropods. So about 1.25 million described living species are arthropods, compared to 1.6 million described living animals on Earth in total, as described. These are numbers that I got from table 29.1 in the book. There's a section in the chapter describing about how it's talking about insects. It's estimated there are way more than this, based upon a study the guy was doing in tropical rainforest trees, looking at all the insects that were coming that he could get from a single tree, and looking at what proportion of them were previously described and those that weren't. So, more than 3/4 of animals, all animals are arthropods. So what are the features of arthropods? Well, these animals have an exoskeleton. So the secdioxazone group in general has that thickened that cuticle outside. In the arthropods, it's thickened with proteins and chitin, which is a waterproof polysaccharide. And the fact that it's a thickened, more rigid cuticle means it can actually act like it acts as an exoskeleton. So, body armor basically. And what that means is that it can move. It has new methods of movement that are not available to animals with thinner cuticle, because it's a place where muscles can attach and pull against to create motion. So, imagine if our bones, rather than being rigid, were all flexible. So in other words, an equivalent here would be having a thin cuticle, and muscles pull against it, or rather than a joint moving, our arm would just bend. That's what this offers. This thickened exoskeleton gives it rigidity that muscles can pull against. So when this muscle pulls, for example, it's going to create movement in this joint down here. And the waterproofing is super important, because this allowed this group to invade and live on land, which were at the time when these first evolved was unexploited niche basically for animals. It allowed many arthropods to go onto land and avoid just drying out basically, because they have this chitin layer within them. But there are many marine, and aquatic arthropods as well. So thick exoskeleton restricts the surface gas exchange. So that means that they had to evolve new ways of breathing, basically, because they can't exchange gas across that thin cuticle anymore. And so we'll talk a little bit about that. Muscles, yes, we talked about that. Arthropods are segmented, and the muscles operate this segment plus pairs of appendages. So we talked about in a previous lecture about how segmentation was so important for adaptability. We talked about crustaceans at the time, for example, because different appendages and segments can evolve different forms that allows a very kind of flexible structure that for evolving for different functions. Okay, different appendages specialized for different purposes. Yes, similar type of thing. Okay, so segmentation and paired appendages is a flexible framework for adaptability. So the earliest of these animals with the jointed appendages were trilobites, which many of you have probably heard about because they're a very common fossilized form if you go to the museum, etc. They were very abundant apparently between 400 and 500 million years ago, but went extinct about 250 million years ago. And one of the reasons why there are so many fossils of them is that this is an animal that is good for fossilizing because they have this thick exocuticle exoskeleton. So it's good for making fossils basically, so there are lots of fossils of them. The earliest group known to possess jointed legs. And you can see here there's lots of segments and all these jointed appendages. Now these are relatively unspecialized, okay, and that some of these differ from the others, but a lot of these are quite similar to one another. So this is a sort of an early form of the arthropods compared to modern day ones that we see today, where a lot of these segments and a lot of these appendages differ between different segments because they've become so specialized. There are 4 major living groups of arthropods that we're going to talk about. Okay, the chelicerates, the myriapods, the crustaceans, and then the hexapods, which includes the insects. Okay, and there's a division between these chelicerates and these other groups in terms of their mouth parts. So the chelicerates are characterized by their mouth parts that are called chelicerae, and these are pointed mouth parts designed for grabbing on to things, but not necessarily for chewing them up. Whereas these other groups are sort of generally or loosely termed the mandibulates, because they have a mandible mouth that is designed or adapted for chewing and biting, as opposed to just holding on to things like the chelicerae. Okay, so the chelicerates include things like sea spiders, a small group, horseshoe crabs, and arachnids. These groups here involve centimillipedes and some relatives of them, decapods like lobsters and crabs, isopods like roly-polies, and some other groups, and then hexapods are mostly insects. Okay, so let's look at these chelicerates. Non-chewing mouth parts, okay, that's what characterizes this group. Over 100,000 species. They consist of these sea spiders, horseshoe crabs, and then the arachnids, okay, spiders, scorpions, harvesters and mites and ticks and things. They have, is that spider? The spider is adorable. Okay, is that a bed bug? It's something along those lines, yes. In terms of that, a mite. This one's less adorable, right? This is a jumping spider here. So they're characterized in general by having 2 body parts. The anterior segments are fused to form a cephalothorax. So in a scorpion, that would be that bit there, for example. And then rear segments, which form the abdomen. The head has 2 pairs of appendages modified into mouth parts. You see the chelicerae that we were talking about. And most of them have 4 pairs of walking legs. Although I thought it was interesting that this pycnogonid they showed, it's got 1, 2, 3, 4, 5, 6, 7 legs. So I guess one of them must have broken off or something. And here you can, this is a good example of these mouth parts on these things. So the sea spiders, they're not actually, these aren't actual spiders. These are different than the arachnids. They're called pycnogonids, about 1,000 species. They're all marine. They live in marine sediments, often in very deep areas. Mostly quite small, but some of them can become quite big, up to 60 centimeters. So, you know, this wide across in terms of their legs, so that's quite big. Horseshoe crabs, there are 4 living species of this. So you don't get these in Europe, on the European side of the Atlantic, but you do get them. They're common along the eastern seaboard here in the US. And so you're probably familiar with seeing either these, or more commonly, probably when you go to the beach, they shed their cuticle. And, you know, that is often washed up on the shore, and you see it there. And these are sometimes referred to as a type of living fossil, which means that their body form has remained pretty much unchanged over the last, well, since the Jurassic, it was saying here. So when you look at a fossil of one of these from 200 million years ago, it's pretty similar to what these things look like today. Common in shallow waters in the US. Now, there's actually an important fishery for these things in the US, because they're actually incredibly valuable, because there's a blood component of them that is used for detecting bacterial endotoxins. And so the medical industry use this for testing stuff. And so there's a fishery for these. They're actually protected in many areas, but they catch them, they keep them in enclosures, and then they bleed them, get their blood, and then put them back alive, well, mostly alive, there is a mortality rate involved, and return them to the ocean. And that's sort of a fairly regulated process. Okay, and with the other Achilles rates, the arachnids, that includes spiders, scorpions, harvestmen or daddy longlegs, and then ticks and mites, okay? Very abundant in terrestrial habitats, okay? So here is one that, you know, can occur in a desert region, somebody was asking about, because this is coming from that group, the arthropods, where they've got that thickened and waterproof cuticle because of the chitin that's in the cuticle, okay? So this group, in general, have a simple lifestyle, meaning that when their eggs hatch, you have miniature adults, so things that look like the adults, as you may be seeing when you look at, you know, spiders hatching in a little web in the tree and stuff. You'll see that they're like little tiny spiders when they first appear. I'm not sure. Okay, sorry. Let's see. Okay, sorry. Somebody was asking about some stuff. Okay, ticks and mites and ticks, about 60,000 species. Many are parasites of plants and animals. This is the time when they're coming out of the woods and getting all over my dog, unfortunately, which is a real pain. Spiders, 50,000 species. Predators with hollow chelicerae that inject venom. So those that are, you know, venomous spiders, you know, are using those mouth parts to inject them. So there are lots of different hunting mechanisms for these. Some have excellent eye vision, such as this jumping spider, and very, very visual predators. And then there are webs. I think it's really, I put this diagram in here just because I find it really interesting. You don't have to learn all these different types of webs, but there's an incredible diversity just in terms of web designs and how spiders have adapted to use webs. So they can produce webs with it's a protein that is produced from the abdominal appendages connected to a special gland. And as that comes out, it solidifies as it comes into contact with air. But yeah, I know that web design is a really interesting area in itself. Okay, so now we're going to talk about the mandibulates. Remember, there were 3 groups of those: the myriapods, crustaceans, and hexapods. So amongst the myriapods, we have the centipedes on the bottom here, and the millipedes. And they're distinguished by the fact that centipedes have a single pair of legs or appendages per segment. In the millipedes, they have each what looks like a segment is actually 2 fused segments. And so each of these has 2 pairs of legs associated with it. Long and flexible, segmented trunk with many pairs of legs, well-formed head with mandibles, being mandibulates, and antennae, and common in like leaf litter and things like that. They're an important recyclers as well. One big difference between these is that, with the centipedes, they're actually have these pincer-like things and can actually be pretty venomous themselves as well. So these things, many of these can bite and give you pretty nasty bites. What attracts centipedes? Moisture. I'm guessing moisture probably, but more likely organic matter like leaf and things like that. Okay. When I was a small kid, I used to actually collect them. I don't know why, but I did. Anyway, crustaceans. Next group we're going to talk about, right? So the second of this group of mandibulates, 67,000 species. And this is the dominant form of marine arthropod today. But they also occur in freshwater and terrestrial habitats. Some of the major groups that we're familiar with are the decapods that include crabs, lobsters, shrimps, crayfish, and so on. Then on land, we have, actually in marine as well, isopods. But this is a group that is common on land that, you see if we pick up a stone, we see these isopods. The reason why they're called isopods, because iso means same, and they have a lot of appendages that are, as an ancestral feature, I guess, are fairly similar to one another, as opposed to highly differentiated like in a crab. And then there's other important groups, amphipods and ostracods, common in marine and freshwater environments. If you scoop a very fine net through some, sort of weed on the bottom of the lake, you'll get a whole bunch of these things. Copepods, one of the most important organisms in the zooplankton in the ocean and in lakes as well. These things occur by the billions and billions in the ocean and the basis of the food web for many larval fish and things like that that feed upon them. And then barnacles, okay, a really big group that was actually Darwin spent quite a long time studying. Okay, crustaceans. Most crustaceans have 3 body regions: a head, a thorax, and an abdomen. They're often covered by the so-called carapace, the head and the thorax area together. The head segments are fused, and there are 5 pairs of appendages in the head. These antennae and antennules are part of those, and maxillipeds, which are sort of feeding appendages. Thoracic and abdominal segments usually have 1 pair of appendages each. So, decapods have also 10 legs. So 1, 2, 3, 4, 5 pairs there. And differentiation between these. And then when you go into the abdomen, you can see that there's differentiation between those and these appendages. So specialization in these different things, in these different areas. And I mentioned the carapace as well. Shell-like feature, so thickened cuticle that covers the head and abdomen regions. Okay, and the last major group we're going to talk about is the hexapods, that includes the insects. Okay, so hexa meaning 6 legs, and this is the largest group on Earth. So we talked about arthropods having 1.25 million species described, and 1 million, more than 1 million of those described species fall within this hexapod group. Okay, abundant and diverse in terrestrial and freshwater habitats, A small number live in saltwater as well. And it includes 3 wingless non-insect groups, plus then the insects. There are actually a couple of wingless insect groups as well that were mentioned, but we'll go through those. And actually here, so yeah, this is showing beetles, Coleoptera, which is the most diverse group of insects, as you probably know. It's a sort of Trivial Pursuit type question. Okay, so this is basic phylogeny of the hexapods. Down here, we'll come on to are the insects, but there are 3 groups outside of this that are not actually classified as insects, although they are hexapods with 6 legs, 3 pairs of legs. The springtails, the coneheads, and the so-called 2-pronged bristletails. So these are pictures of each of these. Now, these are wingless relatives of the insects. So these are wingless hexapods that probably, it's thought, resemble the sort of ancestral condition. So something like this, fairly basic in its form. And they have a simple life cycle. So they hatch, as we had talked about with the arachnids, for example, and some of the other groups, they hatch from eggs in what looks like a miniature adult, basically. So contrast that with the metamorphosis of a butterfly, where you have a caterpillar and a butterfly completely different from one another. So one extreme versus this is the other extreme of that, basically, where these don't go through that. They look very similar throughout their lifetime. What characterizes or distinguishes them from the insects is that they have internal mouth parts, okay? Whereas all the insects have external mouth parts. So that's the distinction between these 3 red groups and this group down here, the insects. Springtails, these guys, can be extremely abundant in soils and leaf litter, up to 200,000 per meter squared, according to the book, okay? So they're not, I don't recognize one of these, you know, as something I see regularly, but nevertheless, they're incredibly abundant if you go and, you know, dig up your vegetable garden or something. So let's move on to the insects then, okay? So just a reminder, we're in the arthropods that have 4 groups. One of them is the chelicerates, the other 3 are the mandibulates. They have mandibles, the myriapods, crustaceans, hexapods. We're within the hexapods of the insects. Their general body plan is that they have 3 regions. There is flexibility within this, but they have a head with external mouth parts, so that is characteristic of the group, and 1 pair of antennae. They have a thorax, okay, here, that has 3 pairs of legs, 1, 2, 3. Sorry, I'm just trying to read the chat there. And an abdomen with no appendages. Okay, so all these legs are attached to the thoracic region. And on the thorax, they also have 2 sets of wings. Now there is modification within that because some have been lost, for example, in some. But the general pattern is 2 pairs of wings. Each antenna has a special organ that's characteristic of the group called a Johnson's organ. It's actually on the 2nd sort of segment of the antenna, and that is adapted as being motion sensitive. So movement in the antenna, for example, can be detected, or movement around it. And they also have a unique gas exchange system with air sacs and tubular channels. So here are the spiracles, these little holes here, that lead into the trachea, which permeate inside the animal. And that's used for gas exchange because many of these animals are terrestrial, And so they can't use gills anymore for gas exchange, and they can't exchange gases across their cuticle because they have a thickened cuticle with chitin and proteins in it, so that they don't dry out when they're on land. So now they need a new method of being able to breathe, and that's how these spiracles come into play. So this is one of the tables from the book. Now, you don't have to, I've sort of said in the comments here, you don't have to go through and memorize this entire table. The important part here is to understand the major groupings within this, and perhaps one or two examples within it, okay? But two insect groups, less derived groups, the jumping bristletails and the silverfish, these are wingless forms of insects, okay? So I said that, you know, in the previous slide or 2 slides ago, we had this general body pattern here. We talked about 2 sets of wings. These are 2 groups, less derived groups, that actually it's thought never evolved wings in the first place, okay? We're lacking of them. So wingless and have simple life cycles. So what I mean by a simple life cycle is that direct hatching into something that looks like a miniature adult, basically. Now, in these other groups, simple is disgusting. Yes, they are. So in this other group, these are known as the pterygoda, and their distinction is that these are the winged insects. Okay, this is the group of insects that are, and vast majority of them, that evolved these 2 pairs of wings. However, as often occurs when we look at these phylogenies, some of these characteristics get secondarily lost over time. So in some groups, the wings have been secondarily lost depending upon the environments that they were evolving and adapting to. That occurred within the lice, fleas, so worker ants, and in some beetles as well. So some of these groups that I've highlighted here, okay? So it's not just this top group here that are lacking in wings. The difference between this group up top and down here is that it's thought these guys never had wings, whereas it's thought with these ones, they had wings but then lost them, okay? Now within the pterygotes, within those insects that possess wings, there is a distinction. The majority of animals are neopterans. And what that means is that they can fold their wings against their body, okay, like a fly would fold its wings down, or a bee or something like that. There are 2 groups that are less derived, and this is thought to be the ancestral condition, the dragonflies and the mayflies, where they cannot do that, okay? If you think of a dragonfly sitting on a blade of something, you know, those wings are sort of like that, and that's the way they stay. So a less derived condition compared to these neopterans. Okay, and we talked a little bit about how these animals go through their development. In these less derived, and when I say less derived, I see more, what I mean is that it's more similar to the ancestral condition. We talked about how these groups here that don't have any wings have direct development, okay, in a similar way to some of the groups that preceded the hexapods. So they hatch, they look pretty similar to the adults, and they grow in size, looking similar each time, going through that ecdysis step, right? Remember when they're shedding that cuticle that cannot grow with them. So hatching of non-pterygote insects looks like miniature adults. Yeah, they increase in size with progressive molts. Now, amongst the pterygotes, amongst those that have wings, this group here have so-called incomplete metamorphosis. So this is where they hatch, and they do go through some kind of a metamorphosis, but it's not as dramatic. So they look fairly similar to the adults, but not the same. So here they've got no wings, and here they have got wings, for example. And with each molt, they're going through a so-called instar. So instar 1, instar 2, etc. in a nymph state. Now they're starting to develop these wings that progressively change into that adult form. So it's a progression. Rachel's the fly, yeah, okay. It's a progression, okay? The most derived groups, in other words, those that have evolved most differently from the ancestral condition, are the, oops, hang about, it seems I'm not plugged in. Oh, there we go, I'm plugged in now. So are the holometabolous insects. So that's within the pterygote. So these are all ancestrally winged organisms. And here they have and here they have complete metamorphosis. Okay, so you know, butterfly being a classic example where you have a caterpillar, so larval stages growing into a caterpillar that looks completely and utterly different from the adult stage that emerges from the pupa. So sort of distinctions between these different groups. So these are called the holometabolous neopterans, okay? Neopterans being this group, you know, this group overall, those with wings. Okay. So, you know, one important point about the insects is they're incredibly speciose, over 100 million species. And what are the reasons for them being so successful, so to speak? So molecular evidence indicates that they began to diversify about 400,000 years ago. And, you know, ancestrally, the arthropods were marine organisms. And so these invaded terrestrial habitat. And we talked about a whole bunch of features of these that, that allowed adaptability. Invasion of land was having that cuticle with the chitin in it that makes them waterproof was really important, because it means that now they're not going to dry out when they come on land. They got the spiracles and the trachea, so they can now breathe on land. Segmentation, paired appendages, which are highly adaptable and flexible in terms of adaptability. And the wings, the evolution of wings. So these were the first organisms to be invading land and also to be evolving flight on land. And that was incredibly important, because it means that they can now disperse across all sorts of different ecological niches and habitats, so that they can adapt to different areas. And this was a whole sort of an open, you know, the Wild West to them in terms of availability of a free niche space and lack of competitors or predators, because these were the first animals to be probably evolved from the dorsal gill elements of crustacean appendages. There's some evidence to support that, or good evidence to support that, from developmental patterns. So in ancestral multi-branched appendage, what's this showing here? Like in a crayfish, they have a gill for breathing in water as a sort of dorsal element of that appendage, which are controlled by the same Hox genes, this PDM gene, for wing development. So it's thought that basically this gill feature sort of changed or adapted. Please can you repeat what you're saying about cutoff for a second or two? The cutoff. What was the cutoff? Sorry. What was I saying about the cutoff? Oh, I cut off. Okay. Can you hear me now? Can you hear me? Yes, good. Okay. The basic story of what I'm saying is that insects were incredibly successful, and so many species, and there's a host of reasons why that is. They were the first animals to be invading land, so there are open niches for them, just as plants were coming onto land as well. So there was a lack of predators and competitors. They evolved wings from the gill elements of marine crustaceans. There's evidence from Hox genes to support that. Wings allowed flight, which means they could then disperse over land and occupy lots of different niches, okay, and adapt into those different areas. Okay, so those are the, plus I also mentioned that they have segmentation with paired appendages. Remember that sort of repeated body pattern allows different regions of the body to be very flexible and adaptable. So you can adapt one area, you know, for one thing, and another, you know, one for walking, one for swimming, or whatever it is, okay? So high flexibility. Okay, now we've got a couple of minutes left over. And does that answer your incomplete metamorphosis? No dramatic change between instars. Complete metamorphosis. Yeah, so the incomplete metamorphosis is more gradual. There is change, you know, when you compare the earliest and the later stages. You know, here there are no wings, here there are wings, but it's a gradual process. Okay, each one is a gradual step. Here, it's a, you know, when you have complete metamorphosis, it's like flicking a switch, basically. Okay, in answer to that question. Hey, let's do one quick, see how many questions we've got time here for. Okay, so if you've got your iClickers going, what is the most diverse group of worm-like ecdysozoans that we talked about at the beginning of the lecture, okay? So remember, we talked about ecdysozoans, the group of protostomes that have to molt a cuticle in order to grow. And a bunch of them are worm-like that have thin cuticles, and then a small number of them, sorry, and a large number of them, another group are not worm-like, okay? They have the jointed appendages. So which is the most diverse group of worm-like ecdysozoans? I got 184 responses so far. Gives you a photo of a phone now. Okay, it gives you a photo now. I don't know what it did. It's set up in exactly the same way. Professor, give me a sec. I need to look. Okay. Okay, we're up to a minute. I'll give you another like 10 seconds. Okay, righty-ho. Got 200 responses now, okay, which is at least 90% of you. So, what do we got here? Can I move that? So it's the nematodes, right? So remember, this is the worm-like ones, 25,000. All these other ones were fairly sort of like small groups. It's distinct from these arthropods that I talked about. Okay, so what was the answer? I think nematodes must have been D, I'm guessing. I haven't got it. What does it say on there? I'm going to mark that as correct because it looks like I'm trying to go back a slide here. The nematodes, yes, exactly, D. Okay, do we have time for another one? I know it's cut-off time. This is a slightly more tricky question, I think. I'll set this one going here. Which of these animal groups is not a mandibulate, okay? Pterogoda, or the pterogoats, horseshoe crabs, millipedes, amphipods, or butterflies. Okay, I'll give you a minute to think about that one. By butterflies, does that include the caterpillar stage? Yes. I should have said Lepidoptera or something. Question's not showing up. It doesn't let me answer. We can't choose an answer. My clicker seems strange. Okay, so I've got 175 responses. I can see that some people are having problems. Refresher, I guess, somebody's saying. First question. I don't know why iClicker is so, it's part of Achieve, that might be it, but what I'll do is I'll go in and check everybody that was on, because it registers when you log in, and I'll check anybody that's logged in, and I'll make sure that you get the appropriate mark, okay? Remember, it's like 0.1 or 0.2%, so anyway, I'm gonna cut it off now. I've got 200 answers, okay? And what have we got here? Okay, so we've got a split decision. Some saying the butterflies and some saying the horseshoe crabs, okay? In terms of mandibulates. Okay, I've got people saying that. So it's actually the horseshoe crabs, okay? Now remember, the pterogoda, these are the winged insects, so these are part of the hexapods. Millipedes are part of the myriapods, amphipoda are part of the crustaceans, butterflies are part of the hexapods. Those were the three groups, the myriapods, crustaceans, and hexapods. So the iClicker sucks, yes. I'll go in and check who was logged into their iClicker and give you the appropriate marks, okay? So don't worry about it. The chelicerates that the horseshoe crabs were in, that was the one group out of the 4 groups of arthropods that are not in the mandibulates, okay? So actually this one here, B, was the correct answer, okay? I know butterflies, when you think of a proboscis on a butterfly, you think, well, that's not a mandible, is it? But the caterpillar stage has got that, right? And they're in the hexapod. That's the main thing. Okay, don't worry. As long as people are logged in, you get the marks. Okay, great. I will send out. I will update the syllabus because that's what people are asking about, and I'll make sure I'll ask Dr. Holtzman about splitting the exam, get advice on that, and I will see you guys on Tuesday. Okay, I'll send out an email with details of the exam, etc. Okay, that for the details that I showed earlier on. Okay, work hard, guys. Focus on the lectures, and have a good weekend. Thank you.
Professor.
Yes.
Quick question. So I know you said you weren't gonna organize it like Dr. Holtzman, but it really did help when she organized it by lecture. Like it helped keep my thoughts in check. So I just kind of want to let you know that.
I know.
If it's possible, I mean, if not, I understand.
It makes it easier. I understand that. You know, the way it would normally be done if you're doing it in class on a sheet of paper is the questions would be randomized.
True, but we could backtrack in person, which makes it a lot easier.
But you can't have the PowerPoint open in front of you there, can you? So whereas you can with this, right? So
I mean, Blackboard
just, you know, it's tricky.
So
you know, it's tricky doing the exams online because there's obviously so much scope for you know, answering questions in a way that you wouldn't be able to if you're in a classroom. Unlikely, I will. Possibly, I don't know, but unlikely. So don't expect it necessarily. Okay.
I have a
Thank you, though.
question, Professor.
Okay, sorry. There's somebody else saying something.
Hi, yes, Professor. I had a question about.
Yeah, you're really quiet. So.
Hello, Professor.
That's better. Yes, you're louder now.
Okay, great. Yeah, I had a question about mollusks and the cuticles. Okay, so what's the difference between like how mollusks use the mantle to form their exoskeleton and then with arthropods and cuticles, right? Is the difference just like the components that they're
Well,
made out of?
the exoskeleton in them is completely and utterly different. So the exoskeleton of a mollusk is calcareous, or well, okay, it's just calcium carbonate or majority calcium carbonate. Okay, the exoskeleton of a shrimp, etc., has got a lot of calcium in it as well, but it's a epithelial layer that is secreting a shell. The shell is completely different in formation in a mollusk, and chemically different, but also it doesn't surround the entire organism. So a gastropod, gastropod can grow, but just adding to its shell all the time. Okay, it doesn't have to escape. It doesn't have to shed a shell to grow. It just has to make a bit more shell to accommodate it. So whereas ecdysozoan is completely surrounded in this, bodysuit basically, that it cannot made of different chemical composition that it has to get rid of in order to grow.
That chemical composition, is that chitin for the arthropods?
So arthropods in the do they all have chitin? So it varies, I think. I think the worm-like ones do not have are not highly chitinized because the chitin is imparting, it's giving it that waterproof layer. So in the group like the insects, for example, they've got lots of that chitin that is not available. It's not there in the worm-like ones, basically. I'm guessing there's probably some, but it's not purely.
No, yeah,
also, they've got
right.
more protein. There's more stuff that's going into it in general. They've got more protein. Some of the decapods have got calcareous skeletons as well. That basically makes it much thicker and much more rigid than those worm-like ones. So the chitin is really imparting the waterproof part to it.
Okay, but a cuticle is present in both the mollusks and the arthropods?
A mollusk you would describe as having a shell rather than a cuticle. Right?
Yeah. Okay, got it. All right, and final question, Professor. So on Achieve, it doesn't look like the pre-lecture quiz for chapter 30 is available to review. Do you think it would be possible that we can go back and review those questions?
So it's they kind of go offline. They become unavailable at the end of the time when they can be done.
Okay.
Right. Otherwise,
right,
people can just carry on, sort of. Yeah.
All right. Okay. Great. All right. Thank you, Professor.
Okay.
Take care.
Professor,
Anybody else out there? Yes.
I have
Somebody.
a question. So I was wondering, since the questions, since you think that the sections will be randomized in terms of chapters, is it possible then, if you're going to do that, just to make it 2 sections instead of multiple sections?
I'll chat to Dr. Holtzman, I think. The trouble with doing that, the whole reason for splitting into sections is that if people have internet crashes, they only have to do a small number of questions again, as opposed to everything. So, the bigger you make those sections, the more questions you'd have to do if you had an internet crash, right?
Yeah, no, I think it's just that a lot of people like the sections how it was last semester, because it was 2 chapters per section. So I think now, since it's randomized, that I don't know if it matters too much how many sections there are.
I'll chat to Dr. Holtzman. It's obviously easier if all the chapters are organized, right?
Right.
That's part of the thing.
I have another question as well.
Sure.
So for the first test, we were told that it would be mostly lecture based. And there were a few questions that were from the textbook and not what was spoken about in lecture. And I was wondering if you've spoken to Professor Spagnolo and if you know if he's going to be doing that again and if we should go back into the textbook to review or if it's going to be more focused on the actual lecture slides.
It's focused on the lecture slides, basically. You know, it's possible that, well, when I say the lecture slides, it's focused on the lecture material. So even if it's not on a slide, it might have been said, right?
Right.
So, or it could be inferred from some way. I mean, you know, when we've talked about it, it's not like, so for example, in the chapter we've just discovered, there's a whole, although I did actually mention it today, but it wasn't in the slides, but there's a whole, you know, sort of section on this guy that did that study that, where he trapped all the insects he could from one giant tree in the tropics or something. And then, you know, then there's a whole page on it, looked at what proportion were identified species and which weren't, and you know the inferences from that, and you can go and work with the data. We didn't cover that in detail. I happened to mention it offhand, but it's not like a big section of the book that we covered, so I'm not going to, that won't be in the exam basically, right?
Right. You just need to know
he's
what
not intending to, he's definitely not intending to put stuff in that he didn't cover in lecture.
No, I wasn't accusing him that he was. I was just like, just more for myself to know how to study for this. It's just what was said in class and what was on the slides, less of us having to just reread the textbook.
Yes, I mean, there's stuff in the textbook that's obviously is what the slides are based upon, right?
Right. But we
What I'm saying is there might be certain sections or topics that are not covered, right? I mean, it's, I don't know how to make it more distinct. It's as best as possible that we can. Do you know what I mean?
So the PowerPoints, basically, the PowerPoints and any notes that were taken during class is the best way, is what you're saying.
And what I say, yeah. So which, you know, your notes would be based upon what is said, right?
Right. Okay. Thank you.
Okay. Okay.
Hi, Professor.
Anybody else out there?
Oh, Professor,
Oh, sorry.
go ahead, go ahead.
Okay, thank you. Hi. I unfortunately came extremely late to class. This is the first time I've done that, honestly, come this late.